Welcome to the Deep Dive. Today we're embarking on a really fascinating exploration, looking deep into the inner workings of the human eye. It's a truly remarkable and incredibly precise system.
It really is. You can think of it almost like the ultimate high-definition camera. Every single component, from the very front lens right back to the sensor at the back, it all has to be perfectly maintained.
Perfectly maintained, yeah.
Yeah.
To capture the world clearly.
Absolutely. And when we talk about that optimal maintenance, we're really focusing on a few key things, like the crystal clear transparency of all the tissues that light has to pass through.
Okay.
And the surfaces, they need to be flawlessly smooth, precisely curved to focus that light correctly.
Right, the fold.
And then there's just the overall structural stability of the eye itself. These aren't just nice to haves, they're absolutely fundamental if you want sharp, clear vision.
That structural stability part.
Yeah.
That's particularly interesting, isn't it? Keeping that perfect globe shape.
And that shape is mainly held by the sclera, that tough white outer layer you see. And also by something called intraocular pressure, basically the pressure inside the eye. And crucially, it's always just a little bit higher than the atmospheric pressure outside.
Right, that internal pressure keeping it inflated, essentially. Okay, so today, our mission is really to dissect these key physiological processes, the ones that keep this amazing system running smoothly.
Yeah, we want to understand not just how a healthy eye works, but also connect those normal functions to the potential disruptions that can actually lead to various eye diseases.
Exactly. So we'll be looking at things like the blood-retinal barrier, that incredibly selective gatekeepers.
And we'll delve into the intraocular fluid, both the aqueous and the vitreous humors, how they're made, how they circulate, what they do.
And intraocular pressure itself, how it's regulated.
Definitely. And finally, we'll take a really detailed look at the metabolism, how all the eye's tissues get their energy, both the ones with blood vessels.
And those clever ones without, like the cornea and the lens.
Precisely. How do they manage?
And as we go through this, we're aiming to really internalize the core facts, right? Not just memorizing terms, but understanding how it all fits together.
Yes. The goal is to see clearly how, disruptions in this normal physiology are often the root cause of so many eye conditions.
And maybe even touch on how knowing this basic science helps develop actual treatments that ophthalmologists use every day.
Absolutely. And also consider how seemingly small things, maybe an issue with the tear film on the surface or subtle changes in the aqueous humor inside, how those can snowball.
Okay, so to kick off our deep dive, let's head towards the back of the eye and examine a really crucial guardian, the blood-retinal barrier.
The blood-ocular barrier.
Sure.
It's the broader term, really, covering both the back, the blood-retinal part, and the front, the blood-aqueous part. It's quite a concept.
Okay.
Imagine the inside of the eye as this highly protected sanctuary. Well, this barrier system is like the sophisticated security, controlling exactly what gets in and out.
Right, not just one gate, but layers of security.
Exactly. It's a series of strategically placed semi-permeable membranes. They meticulously regulate what passes between the bloodstream and the inside of the eye, the ocular cavity. You can sort of think of it in different zones, controlling things differently depending on what that part of the eye needs.
And back in the posterior segment, where the retina is, this barrier is like a multi-layered defense, isn't it?
It is. You've got the walls of the retinal blood vessels themselves. They are incredibly tightly sealed, almost like the blood vessels you find in the brain or spinal cord, very impermeable to lots of things.
Okay, that's layer one.
Then there's Bruch's membrane. It's a thin layer, an extracellular matrix sitting between the choroid.
The vascular layer underneath the retina.
Exactly. Between the choroid and the retinal pigment epithelium, or RPE. That RPE is another crucial layer of cells just behind the photoreceptors.
So the RPE is layer 3.
Pretty much, the RPE cells have tight junctions between them, forming another critical part of this posterior barrier. So yeah, 3 key components working together back there.
Got it. Vessel walls, Bruch's membrane, RPE.
Precisely. Now, if we shift forward a bit to the ciliary region, that's where the aqueous humor is made.
Right.
The barrier there is formed by a 2-layered ciliary epithelium. For anything from the blood plasma to become part of the aqueous humor in the posterior chamber, it absolutely has to get across this double layer of cells.
And there are tight junctions there too.
Yes, particularly between the non-pigmented epithelial cells. Those tight junctions are really important. They act as the main gatekeepers. That's what we specifically call the blood-aqueous barrier, another key control point.
Okay. And then what about the iris? The colored part? Are the capillaries there different?
Yeah. Their structure is a bit more open, you could say. The walls of the iris capillaries are pretty freely exposed to the anterior chamber, that space between the cornea and the iris,
Oh.
through these little folds and gaps called crypts, and also through the iris's spongy tissue, the stroma. So it's a very different level of permeability compared to those super tight vessels in the retina.
And that difference in tightness has real functional consequences, right? Especially for diagnostics.
Absolutely. It's fascinating because those retinal capillaries are so incredibly tight, large molecules floating around in the blood like fluorescein dye.
Which they use in eye exams.
Exactly. Fluorescein is usually bound to albumin, a big protein. It just can't easily leak out of those retinal vessels. And that impermeability is exactly why ophthalmologists can get such clear pictures during fluorescein angiography.
Ah. So the dye stays inside the vessels?
Precisely. It outlines them perfectly, in sharp contrast against the darker retina, like highlighting tiny roads on a map.
That's a great way to put it, a roadmap of the retinal circulation. But that's totally different from the choroid, the layer behind the retina.
Completely different. In the choroidal circulation, that same fluorescein dye passes very freely out of the blood vessels and into the surrounding choroidal tissue. It floods the area.
But it doesn't get into the retina itself.
Normally, no. Even though it leaks out of the choroidal vessels easily, it doesn't usually cross into what we call the subretinal space, that tiny potential space between the actual photoreceptor cells and the RPE layer underneath.
Why not?
Because Bruch's membrane, while let some things through, and crucially that intact RPE layer with its tight junctions, they act as another significant barrier. They stop the dye from leaking from the choroid up into that critical neural space.
Okay, so this highly regulated barrier system, it's essential for clear vision, but it must pose some challenges for getting medication into the eye, right?
You've absolutely hit on a major point. Researchers and doctors deal with this constantly. Because of this blood-ocular barrier, a lot of large molecules think important antibiotics, or even some therapeutic antibodies we might want to use inside the eye. They really struggle to get into the inner eye structures if you just give them systemically.
Systemically meaning like a pill or an IV drip.
Exactly. They just can't easily cross those tight cellular barriers, which is why, sometimes medication needs to be delivered differently, like through direct injections into the eye to bypass these natural defenses.
But there are exceptions. Some things can get through more easily.
Yes, that's an important point. Things that are highly lipoid soluble or fat soluble, they can often permeate these biological membranes more readily.
Why is that?
Well, think of cell membranes as being mostly made of lipids. Fats. So fat-soluble molecules can kind of dissolve into and pass through them more easily than water-soluble ones. The examples given, like sulfonamides and chloramphenicol, both antibiotics, they have this property. Their chemical nature lets them cross those lipid-rich membranes better.
Okay, that makes sense. Now, what happens if this careful system breaks down? If the barrier gets leaky?
Right. If those normally tight junctions between the cells, whether it's the vessel walls, the ciliary epithelium, or the RPE, if they become more permeable, then larger molecules that are usually kept in the bloodstream, like proteins, start leaking out.
And what does that look like?
It leads to this turbid, protein-rich fluid accumulating where it shouldn't be. Depending on where the leak is, you see different things. In the anterior chamber, you get what's called plasmoid aqueous. The normally clear aqueous humor gets cloudy, viscous because of all the extra protein.
Okay.
In the retina itself, the leakage can cause exudates deposits of fluid and protein, which can really mess up the retinal function and impair vision. And if it happens between the choroid and sclera, you can get a choroidal effusion, basically fluid building up there.
And what causes these barriers to weaken or leak in the first place?
Several things can do it. Certain drugs, vasodilators that widen blood vessels, can sometimes make them leakier too. Inflammatory conditions are a big one, things like iridocyclitis, which is inflammation of the iris and ciliary body, or choroiditis, inflammation of the choroid. These can directly damage the barrier cells.
Inflammation makes sense.
Yeah.
Any other causes?
Interestingly, even mechanical forces can play a role. Like if the intraocular pressure drops suddenly and significantly, maybe during eye surgery like paracentesis where they drain fluid, or even just from really vigorous rubbing of the eye, the delicate capillary walls can get stretched, and that can temporarily increase their permeability.
Wow. So it's really delicate balance needed for the eye's internal health. Okay, let's shift focus now to the fluids inside the eye, starting with the aqueous humor. It fills that space at the front, right, between the cornea and the lens.
That's right. Aqueous humor is this transparent, colorless fluid. It's in both the anterior chamber, which is between the cornea and the iris, and also the posterior chamber, that tiny space behind the iris but in front of the lens.
And what does it do?
It has a low refractive index, so it bends light a bit, contributing to the eye's focus.
Yeah.
Its composition is very similar to blood plasma, but with key differences because of that blood-aqueous barrier and active secretion. And it has two absolutely vital jobs.
Which are,
First, it's the nutrient delivery system. It brings glucose, amino acids, oxygen, all the essentials to the parts of the eye that don't have their own blood supply, mainly the cornea and the lens.
right, the avascular structures.
Exactly. And second, it's critical for maintaining the intraocular pressure within that healthy, stable range. That pressure is essential for keeping the eye's shape and its optical properties correct. Think of it like the air pressure in a tire, keeping the eyeball properly shaped so light focuses right on the retina.
Got it. Nutrient delivery and pressure maintenance. And then there's the other fluid, the vitreous humor, filling the big space at the back.
Yes, the vitreous humor or vitreous gel. It's very different from the aqueous. It's a transparent, mostly stagnant gel that fills the vast majority of the eye's volume, everything behind the lens, right up to the retina.
And what's it made of?
It's mainly a network of superfine collagen fibers, type 2 collagen, interwoven with long chains of hyaluronic acid. That hyaluronic acid binds lots of water, giving it that gel consistency.
Are there cells in there?
Very few. Mostly some specialized phagocytes called hyalocytes on its surface, cleaning up debris. Crucially, it's a vascular, no blood vessels. And it's almost entirely water, like 98, 99%.
So it's more about structure than circulation.
Exactly. Unlike the constantly flowing aqueous, the vitreous is much more stable. Its main job is providing structural support and helping keep the retina pressed against the back of the eye, against the choroid.
Okay, so back to the dynamic aqueous humor. It's constantly being produced. Where does it actually come from?
It's continuously formed from blood plasma, specifically by the cells of the ciliary epithelium. That epithelium lines the ciliary body, which is that ring-like structure behind the iris.
The same structure involved in focusing.
Yes, the ciliary muscle is part of it. Now, at the cellular level, it's interesting. There are two layers of this epithelium, pigmented and non-pigmented. They're connected by gap junctions, allowing free communication, letting small molecules and ions pass between them.
Okay, so they work together.
They do. But it's the tight junctions between the non-pigmented epithelial cells, the ones facing the posterior chamber, that are absolutely critical. These form the blood-aqueous barrier, that selective gate we talked about, controlling what gets from the blood into the aqueous humor.
So it's a highly coordinated process, and there are three main mechanisms involved in making this aqueous humor.
That's right. Ultrafiltration, diffusion, and active secretion. All three contribute, but active secretion is by far the most important. It accounts for something like 95% of the total aqueous production.
And that's different from the other two.
Yes. Ultrafiltration and diffusion are passive, driven by pressure or concentration differences. Secretion is active. It requires energy from the ciliary epithelial cells. They use their metabolic power to actively pump specific ions and molecules from the blood into the posterior chamber. Water then just follows passively because of the osmotic gradient created.
And we know some of the key things being actively pumped, even if the whole mechanism isn't fully understood.
Yes, we do. We know the non-pigmented ciliary cells actively transport a watery fluid that's especially rich in sodium ions, ascorbic acid, vitamin C, a key antioxidant, and bicarbonate ions, plus other specific things into that posterior chamber.
What's driving that?
A key player is the sodium-potassium ATPase pump. It sits on the membrane facing the posterior chamber and pumps sodium out of the cell into the chamber. This creates an electrochemical gradient that pulls other ions and water along. Another enzyme, carbonic anhydrase, helps make bicarbonate ions, which also get transported out with sodium, further driving fluid movement osmotically. It's very regulated, very energy intensive.
Okay, so the aqueous humor isn't just filtered plasma, it's actively modified. What does its final composition look like then?
It's basically a carefully controlled, dilute solution. It has many of the small diffusible bits from blood plasma, electrolytes, glucose, amino acids. But it also has those actively secreted things, like the high levels of ascorbic acid.
And because of the barrier in the outflow?
Right. Because it's tough for many larger things to get in across the barrier, and relatively easy for fluid to get out through the drainage channels, many components are actually at lower concentrations in the aqueous compared to blood plasma. The exceptions, of course, are those things actively pumped in, like ascorbic acid, where the concentration can be much higher than in blood.
That makes sense. Easier out than in for many things. But there was one notable exception mentioned, an excess of lactic acid in the aqueous compared to blood. Why is that?
Ah, yes. That higher lactic acid level is mainly a direct result of the lens doing its metabolic work.
The lens produces it.
Exactly. As the lens breaks down glucose for energy, especially through anaerobic pathways, which we'll get to, lactic acid is a major end product. This lactic acid then diffuses out of the lens into the surrounding aqueous humor. So you find significantly more lactic acid in the aqueous than you do in the bloodstream. It's a neat example of how one tissue's metabolism directly impacts the fluid bathing it.
It really shows how interconnected everything is. Now, this aqueous humor isn't static, it circulates constantly. Why is that circulation so vital?
Absolutely critical for two main reasons. First, as we said, it delivers nutrients, glucose, amino acids to the cornea and lens, which have no blood supply. And just as importantly, it carries away their metabolic waste products, like that lactic acid. Without this flow, they basically starve and fill up with toxins.
Okay, nourishment and waste removal. And the second reason?
The second is regulating intraocular pressure, the IOP. That constant production and drainage of aqueous humor is the primary way the eye maintains a stable, healthy pressure. The balance between production rate and outflow rate is absolutely crucial. If that balance gets disrupted, IOP can go too high or too low, and both can seriously damage vision.
So what's the main route this aqueous takes? After it's made by the ciliary epithelium, where does it go?
Okay, the primary path, the main flow, goes like this. It's produced in the posterior chamber by the ciliary epithelium. Then it flows forward through the pupil, the opening in the iris, and into the anterior chamber, that space between the cornea and the iris. From the anterior chamber, it then leaves the eye through a specialized drainage system.
Where does that located?
It's right at the angle where the cornea and the iris meet the anterior chamber angle. This drainage system is pretty complex. It starts with the trabecular meshwork, which is like a sieve.
Okay, a filter.
Then it flows into Schlem's canal, a circular channel that collects the aqueous. From there, it goes into collector channels and then into the aqueous veins. These veins merge with the episcleral veins on the outside surface of the sclera, returning the fluid back into the general blood circulation.
That sounds like a pretty direct route out.
Mm-hmm.
But you mentioned there's also a secondary alternative exit pathway.
Yes, there is. It's called the uveoscleral outflow. It's less conventional, you could say.
How does that work?
Instead of going through the trabecular meshwork in Schlem's canal, some of the aqueous humor actually permeates through the tissues of the ciliary body itself into the suprachoroidal space, that potential space between the choroid and the sclera.
And then where?
From there, it eventually drains out through the sclera itself, or maybe into blood vessels in the choroid and episclera. This pathway is interesting because it's less dependent on pressure than the trabecular route.
And it handles less of the total drainage.
Typically, yes, usually around 10–20% in a healthy eye, but it becomes clinically quite important, especially in glaucoma, if the main trabecular route is blocked or compromised. Some glaucoma meds actually work by boosting this uveoscleral outflow to help lower the pressure.
Interesting. So the eye has a backup system to some extent. Okay, let's dig deeper into intraocular pressure, IOP. We know it's key for shape and optics. What are the main things controlling it?
Well, as the source really stresses, the number one factor determining IOP is that dynamic balance, that equilibrium between how fast aqueous humor is being produced by the ciliary body.
Right, the inflow.
And how efficiently it's being eliminated, drained out of the eye, mainly via Schlem's canal and those outflow channels. It's like a sink. If the tap production runs faster than the drain outflow can handle, the level pressure goes up. If they match, it stays stable.
Simple analogy, but it works. And while that's the main thing, there are other factors causing short-term ups and downs.
Yes, definitely. Besides that primary balance, other things can cause temporary or transient shifts in IOP, like the amount of blood volume within the choroid and the vitreous. If that volume increases, it can briefly raise the pressure inside the rigid scleral shell.
Makes sense.
Also, the tone of the extraocular muscles, the ones that move your eyeball, when they contract forcefully, they can put a little squeeze on the globe, briefly affecting the pressure.
But for the longer term, sustained changes in IOP, it really goes back to those two things, aqueous formation rate and outflow resistance. And clinically, it's usually the outflow part that's the problem.
That's absolutely right. While changes in production can happen, prolonged, significant IOP changes are most often caused by problems with the outflow increased resistance to drainage. That intricate trabecular meshwork and Schlem's canal system, it's just more vulnerable to things going wrong and blocking the flow.
So what kind of things can block or slow down that outflow?
Well, a persistent rise in IOP can happen if there's increased resistance getting into Schlemm's canal. This could be due to gradual hardening or sclerosis of the trabecular meshwork itself. It just gets less porous over time.
Okay, wear and tear almost.
Kind of. Or it could be a physical blockage. Inflammatory debris cells, proteins, exudates can clog it up, or abnormal tissues like scar tissue or new blood vessels can grow over the angle and block the drain.
Neovascularization, right.
Yes, and less commonly, the pressure after Schlemm's canal could be high. If the pressure in those episcleral veins that the aqueous eventually drains into is elevated, it creates back pressure, making it harder for aqueous to leave Schlemm's canal.
And any of these leading to high IOP can result in
Glaucoma. That damagingly high pressure is the hallmark of glaucoma, caused either by reduced outflow or much less commonly by overproduction.
And even if that main drain, Schlemm's canal, is blocked, the uvascleral pathway still offers some escape route, right? Just maybe not enough.
Exactly. That's an important point about the eye's sort of built-in safety net. If the trabecular pathway is compromised, the uvascleral route can still drain some fluid, but as we said, its capacity is much lower. So while it helps a bit, it often can't fully compensate if the main drain is significantly blocked, and the pressure will still tend to rise.
So any inefficiency in those drainage channels is key for gradual rises or sudden spikes in pressure. Let's quickly revisit those other temporary effects on IOP you mentioned earlier.
Right, so transient changes in the hydrostatic pressure in the eye's capillaries can have a brief effect. Also, remember we talked about increased capillary permeability leading to protein-rich plasmoid aqueous.
Yeah, the leaky barrier.
Well, that extra protein in the aqueous can temporarily increase its osmotic pressure compared to the blood. This can actually draw more fluid into the eye, potentially raising the IOP for a bit. And this effect is worse if the drainage channels are already a bit clogged, like during eye inflammation.
And changing the blood's own osmotic pressure can affect IOP too.
Yes, another fascinating link. If your blood plasma becomes hypotonic, meaning less concentrated, more watery compared to the eye fluids, water can move from the blood into the eye, potentially bumping up the IOP temporarily. That's the idea behind the water drinking test used sometimes for glaucoma suspects. Conversely, if the blood becomes hypertonic, more concentrated, water gets drawn out of the eye, lowering the IOP. That's why doctors sometimes use hyperosmotic agents like glycerol or mannitol intravenously to quickly bring down dangerously high IOP in emergencies.
Makes sense. And given the sclera isn't very stretchy, adding extra volume inside the eye must spike the pressure quickly.
Exactly. Because the sclera is tough and relatively inelastic, any significant increase in the volume of stuff inside the eye, like a large vitreous hemorrhage bleeding into the gel, can cause a rapid, substantial rise in IOP. There's just nowhere for the pressure to go.
And finally, blockages within the aqueous circulation path itself, even before the drain, can also seriously raise IOP.
Yeah.
Two key spots were mentioned.
Yes, the source highlights two main choke points where blocking the normal aqueous flow can dramatically hike up the pressure. First is at the pupil. Flow from the posterior chamber where it's made to the anterior chamber can get blocked. Pupillary block.
That happened.
It's more likely in eyes with shallow anterior chambers where the iris is naturally closer to the lens. Sometimes the iris can actually stick against the front of the lens, stopping aqueous from getting through the pupil.
And the pressure builds up behind it.
Precisely. Pressure rises in the posterior chamber, making the iris bow forward. That's called iris bombée. This bulging iris can then slam shut the drainage angle in the anterior chamber, causing a sudden painful attack of primary angle closure glaucoma, sometimes called relative pupillary block initially.
Ouch. And the second critical blockage point.
Right at the drainage angle itself, where the aqueous is supposed to leave, the trabecular meshwork can get physically obstructed.
How?
Usually by adhesions forming between the peripheral edge of the iris and the cornea, literally sticking the iris over the drain. This can happen in angle closure, like we just discussed, or after chronic inflammation leaves scar tissue fibrosis there, or if abnormal new blood vessels grow across the angle neovascular glaucoma. These are all types of secondary glaucoma because the angle block is caused by some other underlying condition.
Okay, so a whole host of factors can push IOP up or down, short-term or long-term. But it really hinges on that production versus outflow balance, what's considered the normal, healthy range for IOP.
The generally accepted normal range is between 10 and 20 millimeters of mercury, usually written as millimHg. But it's important to remember that's a statistical average. What's normal for one person might not be for another. Depends on other things, too.
And how do doctors actually measure this in the clinic? Can't stick a needle in routinely.
No, definitely not. The most direct way is manometry, inserting a fine needle connected to a pressure gauge right into the anterior chamber. Super accurate.
But invasive.
Highly invasive, yeah.
Yeah.
Carries risks like infection. So it's really only used experimentally, mainly in animals or very specific research, not for your regular eye checkup.
So what do they use clinically?
The standard is tonometry. There are different kinds, but they basically work either by measuring how much the cornea resists being indented by a known weight or how much force it takes to flatten a specific area of the cornea. The gold standard is usually considered Goldman applanation tonometry. They numb your cornea with the drops, then a small, flat-tipped prism gently touches the cornea. The machine measures the force needed to flatten a standard 3.06 millimeter diameter circle. That force reading is then converted into millimeter Hg based on calibrations done against manometry in animal eyes years ago.
And there are non-contact ones too, the puff of air test.
Yes, non-contact tonometers use a quick puff of air to flatten the cornea, and they measure how the cornea deforms to estimate the pressure. Tonometry is absolutely essential in eye exams, especially for screening and monitoring glaucoma, where high IOP is often the main treatable risk factor.
Right. Okay, we've covered barriers, fluids, pressure. Fascinating stuff. Let's switch gears now to metabolism. How do the eye's tissues actually fuel themselves? The source divides them into vascularized and non-vascularized. Let's start with the ones with a blood supply.
Okay. For the eye tissues that do have their own blood vessels, particularly the uveal tract, which includes the iris, ciliary body, and choroid, their general metabolism is pretty much like other well-vascularized tissues elsewhere in your body. They get a constant supply of oxygen and nutrients delivered directly by blood flowing through their capillaries, and waste products are efficiently carried away by the same system.
And they need quite a bit of energy.
They do. They have relatively high metabolic demands to support their functions, controlling the pupil, making aqueous humor, adjusting focus, nourishing the outer retina, so they rely heavily on that direct blood supply.
Makes sense. Built-in plumbing. Now the tricky ones, the non-vascularized tissues. The cornea at the front and the lens behind the iris, they need to be transparent, so no blood vessels. How do they survive?
Exactly, that's their unique challenge. They've evolved these really clever ways to get energy and nutrients while staying perfectly clear. They mainly rely on metabolizing carbohydrates, mostly glucose, which they get from the aqueous humor bathing them. They use pathways like glycolysis and other autooxidative systems to generate the energy they need for basic cell maintenance, repair, and their specific jobs related to vision. Having no blood vessels is absolutely key, for their transparency vessels would just scatter light all over the place.
Let's focus on the cornea first. The clear window at the front, what about its metabolism?
The cornea, even though it's exposed to the outside world, actually has fairly modest energy needs. Its main jobs metabolically are turning over and repairing its surface cells, the epithelium.
Right, the outermost layer.
And, crucially, actively maintaining its amazing transparency. And that transparency, as we've touched on, heavily depends on keeping the cornea in a state of relative dehydration, especially the stroma, the thick middle layer.
How does it stay dehydrated?
There are active pumps, mainly in the endothelium, that single cell layer on the inner surface, facing the aqueous humor, and also in the epithelium. These pumps constantly work to move fluid out of the stroma and into the aqueous humor. If the metabolism of those endothelial cells falters, or if the cornea's membranes become too permeable from injury or disease, fluid builds up,
leading to swelling, corneal edema.
Exactly. The cornea swells, gets hazy or cloudy, and vision gets significantly blurry. That endothelial pump is vital.
And structurally, the cornea is very different from the sclera, the white part, even though they're connected. How does that relate?
Yes, the key difference physiologically is how their collagen fibrils, the main structural proteins, are arranged. In the cornea, the fibrils are incredibly uniform in size and spacing, laid out in this highly ordered latticework pattern within a ground substance.
Like a perfect grid.
Pretty much. And that regular arrangement, with the spacing smaller than the wavelength of light, is what minimizes light scatter and makes it transparent. The sclera, on the other hand, has thicker, more irregularly arranged collagen fibers, all interwoven strong, but opaque.
And the cornea has those active cell layers.
Right, the epithelium on the outside, endothelium on the inside. These cellular membranes actively control fluid and solute traffic. The sclera doesn't have that kind of active regulation.
So no blood vessels. Where does the cornea get its oxygen and nutrients from? Three main sources mentioned.
That's right, it's quite resourceful. First, oxygen comes directly from the air. The surface epithelium is very active in taking up atmospheric oxygen. If you deprive it of oxygen, say in a nitrogen atmosphere, lactic acid builds up quickly, showing how much it relies on that aerobic process.
Okay, oxygen from the air. What else?
Second, it gets solutes, glucose, amino acids, other nutrients diffusing in from the tiny blood vessels at the edge, the paralimbal capillaries, where the cornea meets the sclera. You see the importance of this because the peripheral cornea is often more resistant to certain degenerations.
Right.
And third, the aqueous humor itself, bathing the back surface, is a major source of nourishment, providing glucose and other essentials. Interestingly, you can cut off the limbal blood supply, and the cornea might stay clear thanks to the air and aqueous. You can replace the air with nitrogen, and it might stay clear if the aqueous is normal. But if you cut off both the limbal supply and the aqueous nutrition, then it will become opaque.
So it needs at least one good source. And metabolically, it favors aerobic glycolysis.
Yes, that's a notable feature. Even though it uses oxygen from the air, the cornea heavily relies on glycolysis breaking down glucose to pyruvate even when oxygen is present. That pyruvate can then be fully oxidized in the mitochondria. This high rate of aerobic glycolysis might be related to minimizing byproducts that could scatter light.
Interesting. Okay, now let's shift to the lens. Also non-vascularized and completely surrounded by aqueous humor. Its situation seems even more constrained.
It is. The lens is entirely encapsulated and gets all its nourishment solely from the aqueous humor around it. Fluid, nutrients in, waste products out, it's all tightly regulated by the semi-permeable lens capsule, the outer layer, and the metabolic activity of the subcapsular epithelium.
The epithelium is just under the capsule.
Yes, a single layer of cells right under the anterior part of the capsule. And just like the cornea, if that capsule gets damaged, the lens tends to soak up fluid from the aqueous, swell up, lose transparency, and that's how you get a cataract.
So that capsule and epithelium are crucial gatekeepers.
Yeah.
And there's active transport going on there too.
Absolutely. That subcapsular epithelium is actively pumping ions, amino acids, glucose between the aqueous and the lens interior. It maintains the specific internal environment the lens needs. The capsule itself lets water, electrolytes, and small molecules through, but blocks larger proteins. And permeability isn't uniform. The back part of the capsule is generally more permeable than the front, and overall permeability tends to decrease with age.
What about oxygen for the lens? Does it get much from the aqueous?
Not a lot, no. The aqueous has some dissolved oxygen, but the lens itself is considered to be in a relatively low oxygen or hypoxic environment. Because of that, its main metabolic processes are largely anaerobic. They don't need oxygen.
So it relies heavily on glycolysis.
Very heavily. The lens is packed with enzymes for anaerobic glycolysis, breaking glucose down into pyruvate. And then that pyruvate is mostly converted into lactic acid and water, which, as we said before, then diffuses out into the aqueous humor, raising the lactic acid level there. You see high lactic acid in aqueous from an eye with a lens, but not if the lens has been removed, a phakic eye.
And the source mentioned glutathione and ascorbic acid vitamin C being really important for the lens.
Critically important. They're powerful antioxidants. They're thought to work together in an internal defense system, protecting the lens proteins and lipids from oxidative damage from things like reactive oxygen species. That kind of damage is a major factor in cataract formation.
Are they found in high amounts?
Yes, remarkably high concentrations. Glutathione, especially in the outer layer, the cortex, and ascorbic acid is actively pumped into the aqueous by the ciliary body, and then the lens takes it up, concentrating it. Significantly, in cataractus lenses, the levels of both glutathione and ascorbic acid are way down, sometimes almost gone, really highlights their protective role.
And where does most of this metabolic action happen within the lens? Is it all active?
No, it's not uniform. Most of the metabolic activity is concentrated in the outer part, the cortex, and especially in that subcapsular epithelial layer at the front. The central part, the older nucleus, is much less active metabolically. It's relatively inert. It's like the younger outer cells are doing the work to maintain the whole structure.
Okay. And the source went into a bit more detail on how the lens uses glucose, several pathways involved.
Yes. Glucose from the aqueous is the main fuel. It gets processed through a few interconnected pathways. The main one for energy is glycolysis, the EMP pathway, which, as we keep saying, is mostly anaerobic in the lens.
Right.
Then there's the pentose phosphate pathway. Its main job isn't making ATP, but generating NADPH, a crucial molecule for antioxidant defense, especially for recycling that glutathione and also making building blocks for things like nucleic acids and proteins.
And the third one, The TCA cycle.
The TCA cycle, or Krebs cycle, this one does need oxygen, so it's less prominent in the lens overall because of the low oxygen levels. But it's very efficient at making ATP when it can run. It happens more in the epithelium, which probably gets a bit more oxygen. All these pathways generate ATP, the cell's energy money.
And most of the glucose entering the lens gets funneled down one main initial path.
That's right. The vast majority, maybe 90–95%, as soon as it enters a lens cell, gets phosphorylated to glucose 6-phosphate by an enzyme called hexokinase. This basically traps the glucose inside and commits it to metabolism.
What happens to the rest?
A small amount gets diverted into something called a sorbitol pathway, started by the enzyme aldose reductase. Normally this is minor, but if glucose levels are really high, like in diabetes, this pathway becomes much more active. Sorbitol builds up inside the lens cells, draws water in osmotically, and that stress is thought to be a major contributor to diabetic cataracts.
Okay, so most glucose becomes glucose 6-phosphate. Then where does that go? Mostly glycolysis.
Exactly. About 80% of the glucose metabolized goes down the anaerobic glycolysis EMP pathway. Breaks it down to pyruvate, generates some ATP and NADH. Given the low oxygen, this is the main energy source for most of the lens fiber cells.
And the pentose phosphate pathway, about 10%, more for protection and building blocks.
That's a good summary. Roughly 10% goes that way.
Right.
Super important for making NADPH, which the lens needs constantly to regenerate its antioxidant defenses, particularly glutathione, and it makes the 5 carbon sugars needed for synthesizing DNA, RNA, and other bits for proteins and membranes.
And finally, the TCA cycle. Only about 3% of glucose, but efficient ATP production where possible.
Correct. Only a tiny fraction, maybe 3%, gets fully metabolized aerobically via the TCA cycle because oxygen is so scarce. But even that small amount is significant for ATP production, maybe contributing 25% of the lens's total ATP, with glycolysis providing the bulk, around 70%. And again, this aerobic part is likely more active right at the front, in the epithelium.
It's just incredible how these tissues manage without blood vessels. These intricate, specialized metabolic strategies are key to their function and transparency. Okay, let's try to pull this all together now as a summary of these key physiological processes.
Right. So as we've seen, the healthy eye relies on this really delicate, beautifully balanced interplay of many processes. You've got the blood-ocular barriers acting as selective gatekeepers. You have the constant production, circulation, and drainage of the intraocular fluids, aqueous and vitreous, providing nutrients, removing waste, maintaining shape and pressure.
That pressure regulation itself, the IOP, being so critical.
Absolutely. And then the unique metabolic strategies of all the tissues, vascular and avascular, keeping the cells alive, and for the cornea and lens, keeping them transparent. And we shouldn't forget the tear film on the outside, its homeostasis, is vital for the ocular surface health, preventing discomfort, inflammation, infection risk.
And just recapping some specific mechanisms. Aqueous formation involves both passive ultrafiltration in the ciliary processes, where capillaries are leaky,
and active secretion by the ciliary epithelium, driven by things like the sodium-potassium pump and carbonic anhydrase, moving ions and water into the posterior chamber.
Right, and corneal transparency comes from that combination of relative dehydration, no blood vessels or cells in the stroma, and that super regular lattice arrangement of collagen fibrils, smaller than light's wavelength.
Exactly. And the lens crucial for refraction, focusing, accommodation, and absorbing UV light stays clear through a similar strategy. Regular packing of its fibers, thin epithelium, no blood vessels.
Plus those protective antioxidants.
Yes. High glutathione levels protecting against oxidative damage, the sodium-potassium pump maintaining water balance, chaperone proteins like alpha-crystallins preventing protein clumping, and its heavy reliance, about 85%, on anaerobic glycolysis for energy in that low oxygen setting.
It really drives home how understanding these fundamentals is absolutely crucial. It's a foundation for grasping eye health, but also what goes wrong in disease. It gives you such a deeper appreciation for just how complex, how resilient, and frankly, how ingenious the eye really is.
It really does. And thinking about this delicate balance, this intricate machinery, it does raise some interesting questions for you, the listener, to think about. For instance, considering how interconnected everything is, what might be some less obvious ways that external factors, our environment, maybe our lifestyle, could subtly impact these systems and affect our eye health over time?
That's a great point.
And looking ahead, as our understanding of these physiological processes gets deeper and deeper, down to the molecular level, how might that knowledge lead to completely new ways, maybe novel therapies, for tackling the eye diseases that still cause so much vision loss?
Really fascinating directions to consider. Well, thank you for joining us on this deep dive into the intricate and absolutely essential physiology of the human eye. We hope it sparked your interest, and we definitely encourage you to keep exploring the captivating world of ocular science.